Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our expert guest, Dr. Geoff Miller. Listeners, Geoff is lab manager at the Sports Medicine Research Testing Laboratory, or SMRTL for short. And today we are going to talk more about SMRTL, Geoff's research, and his experiences as a former PCC Research Fellow. So, Geoff, welcome to the show. How are you doing today?
I'm doing great. Thank you for having me.
Well, thanks so much for joining us. Can you maybe just start by telling us more about SMRTL and your role there?
Sure. So SMRTL, or as you said, Sports Medicine Research and Testing Lab, we're located just south of Salt Lake City, and we're one of two WADA-accredited anti-doping labs in the United States. The company was founded in 2003, initially associated with the University of Utah. Then in around 2008, we became an independent company. And in 2020, right as COVID was setting in March, we moved into our current facility, which is, as I mentioned, south of Salt Lake City in South Jordan, Utah. My role at the company is twofold. So I have one foot in the routine testing area of the lab, which is responsible for the analysis of anti-doping urine and blood samples. Unfortunately, I don't get to get into the lab too much to work at the bench anymore. So primarily, I'm responsible for the oversight of the EPO, human growth hormone, HCG, and blood testing. On that end, I'm also the director of our Laboratory Athlete Passport Management Unit, or APMU. So that responsibility includes review of athlete biological passports, blood and steroid data, and then liaising with testing authorities and results management authorities to provide testing recommendations based on the data that's in the passport. The other role that I have in the laboratory is related to general research projects, where it's primarily related to design and internal oversight of mostly what we call our clinical research studies. A lot of these projects relate to drug studies in a human population, where we'll study urinary excretion patterns and look at things like fundamental kinetics of substances in blood. Ideally, that's a 50-50 split, but it never works exactly that way. So just depending on the day of the week or the time of year, I may be all in on the routine anti-doping work. I also may be all in on research. But having the ability to work in both areas is excellent because there's always something going on to keep me busy.
Fantastic. And it sounds like you've got a lot of different responsibilities that you're juggling there. And you mentioned that a big part of your role is oversight. So can you tell us a little bit more about the team that you're helping oversee? How many people work within the routine testing side of things as well as the research side?
In the routine testing side, we have approximately, I think we're over 50 employees right now. And my team specifically, there's about 4 or 5 of us that work in that area of EPO, human growth hormone, and HCG testing. For the research end, we are quite small. We have about 2 or 3 individuals here that work primarily in the research department.
Oh, cool. And I know, Geoff, you have an interesting story about how you found your way to anti-doping. So can you tell us how you first got interested or maybe introduced to this field?
If I start a little bit further back, I grew up in the 90s and the 2000s. I played a lot of sports and I was a big fan of sports as well. So I was a kid and a teenager during the major doping scandals that we're all familiar with. Lance Armstrong in the Tour de France era was obviously a big story at the time. And I also vividly recall the Balco scandal in the United States and then the congressional hearings that were related to steroid and PED use in baseball. So at the time, those were very big stories in the news. When I finished college, I went to the University of Utah for graduate school, and the work actually had nothing to do with anti-doping. I studied in the pharmaceutical chemistry department at the College of Pharmacy. Most of the work that we did was focused on cancer research and even more focused on peptide and protein chemistry and drug design. During that time in graduate school, as part of the weekly seminar series, our current director at SMRTL, Dr. Eichner, he gave a seminar on the science and the chemistry behind drug detection in sports. I attended because it sounded interesting. It was the first time that I had really heard of the merging between sports and chemistry, which were two of my interests at the time. So I basically went up to him after the seminar and asked if I could work as a summer intern. That happened in the summer of 2013. And I kept in touch working part-time with the lab until I completed my PhD at the end of 2014 and beginning of 2015. And ultimately, I never left the lab after that.
Oh, that's fantastic. And I understand that you were awarded a PCC fellowship, of course, in 2014. So can you talk a little bit about what the fellowship experience was like for you?
Sure. So during the 2 years of the fellowship, we made it a major point of emphasis for me to learn as much as possible about anti-doping and about laboratory operations as possible. While a major focus of that fellowship is research, just as important is learning the ins and outs of the anti-doping field and especially the anti-doping laboratory. As I've come to learn over the years that I've been here, the more you know about the field, the more you realize where there are certain needs and certain holes that need to be filled in. Identifying where those needs are can help drive future research projects. So it was a great experience that I had in, I think it was 2015, 2016 with the fellowship. We had a lot of freedom here to learn laboratory operations during downtimes in research or off times in the research as well. So a big part of it was learning both the laboratory operations and then becoming familiar with the type of research that we do in this field.
Definitely. So was it tough making that transition into anti-doping when you didn't necessarily have a background in that part of chemistry?
Some parts of it are still tough. There's still some areas of the lab where I'm certainly not an expert. So you're always learning, which is great about this field. The transition was fine because it was something that I was passionate about. So I didn't have a problem coming into work, coming in to do the research projects. It was always fun for me. So the transition was easy in that sense. People that have been in this field for 40 or 50 years, they're still going to tell you that they're still learning and still adapting. Everybody's constantly transitioning in this field as new drugs come out, as new testing methods, new analytical methods. As things improve, everybody in this field has to adapt.
Absolutely. And that's something I've heard from a lot of people in anti-doping. And one of the things that they really enjoy about the field, this kind of constant learning, this constant adapting to a dynamic environment in anti-doping. So can you tell us a little bit more about what you've really enjoyed or found most beneficial about the fellowship with the PCC, maybe about working with the PCC as well?
Working with the PCC specifically, I think one of the more stressful things that a lot of scientists will tell you about their job, especially research scientists, is trying to get funding for their research projects. I've never worked professionally in academia, but I do know that the funding rate for research grants is not great. That's fairly low, but that's not true with the PCC. I don't know if there's less applicants or more targeted research, but what we've learned is they're very responsive to grants and to funding opportunities. And that's something that I've enjoyed and found very beneficial about working with the PCC. Their programs too with the microgrants are outstanding. As scientists, we get a lot of ideas for quick and dirty studies that we know won't require 1, 2, 3 years worth of work, something that we may want to get done in 2 to 3 months or 6 months that won't require hundreds of thousands of dollars. So the fact that we can get a microgrant funded within a week to quickly do a study is a very novel idea to the PCC. We try to be proactive in the field of anti-doping, but sometimes you do find yourself being reactive. So if we know that there's new drugs out or we learn about new methods that athletes may be using to try to skirt the tests, if we have a quick idea that we think may address that issue, that's something that can be very quickly and easily funded.
Absolutely. So maybe can we take a step back here and can you give us an overview of just the kinds of research that you're doing or maybe the methods that you're using in your work?
So that will depend on the project that we're doing. Most of the research that I'm involved with, very simply put, involves giving drugs to human volunteers and looking at urinary excretion patterns, looking at blood kinetics, blood levels of drug and metabolites at various times and at various stages after dosing. So in terms of the methods, that will depend simply on the drug that we're studying. We've done autologous blood transfusion studies here that have involved whole blood analysis on a Sysmex. We've done EPO studies that involve that as well, but also involve gel electrophoresis. One of the areas that I'm a little bit less familiar with is one of the more commonly used areas in this lab. Most of the work we do involves mass spectrometry. So in some of our drug studies, whether it be testosterone or other drugs, we'll get help from other areas of the laboratory with the analysis and instrument operation. So we really use every tool that we have at SMRTL for our research projects.
Fantastic. And I know when you're doing research with humans, there can be some challenges and recruitment is often one of those things in clinical research. So is this something where you're having to go out in the community and try to recruit people yourself? Or is this something where you've got a pool of people that you work with regularly?
So it's a little bit of both. We've been doing this now for 7 or 8 years. So I don't want to say identified a pool, but there's a lot of people that are interested in doing these types of studies. You're giving people performance enhancing drugs and you're paying them for it. So we do get a lot of interest from that end. So we do get a lot of repeat volunteers for various studies. Before we moved to South Jordan, we were located near the University of Utah. We knew at that time there were a lot of graduate students, specifically in the sciences, that were very interested for several reasons. One, because it was an introduction for them to clinical research. Them being graduate students in science, they were interested in the science of it as well. So we have picked up a lot of volunteers from that area. And then recruitment certainly at all times is still difficult. Some of the studies we do may only involve 5 to 10 volunteers. For the bigger ones where we're using 20 to 30 volunteers, those can be a little bit more difficult to recruit and certainly require a lot more time.
Definitely. And you mentioned that, of course, the methods and what you're doing in the research lab can depend on the project that you're working on. So, Geoff, is there a particular project that you have in progress now, perhaps, that you are just the most excited about that you want to give us some more details about?
Yeah. So right now we only have one administration study ongoing, and that actually involves, we're looking at a urinary and blood excretion pattern of HCG, of chorionic gonadotropin after sub-Q administration. Right now, we've enrolled 10 participants in the study, and we hope to be finished with patient visits by the end of August 2022, which is coming up soon. The data that we've generated so far has been excellent and actually very, very interesting. But unfortunately, that's all I can really say about it at this time. We need to wait until we finish enrollment and visits from all the volunteers in order to make a sound interpretation of the data.
Absolutely. Well, you've piqued my interest. I'm sure the listeners as well. Can you tell us at least why you chose to look at HCG?
One thing we've noticed in anti-doping is there's a lot of drugs that we know how to detect, but we don't know too much about urinary kinetics or urinary excretion kinetics of these drugs. When a lot of pharmaceutical companies initially introduced a lot of these drugs, these types of studies weren't necessarily done because clinically there isn't a huge use for it. Most of the pharmacokinetic data generated, and rightfully so, is in blood, but that doesn't translate directly to what we do in anti-doping. Most of the testing we do is in urine. So what we want to see is what do urinary concentrations look like after certain doses, how many hours, how many days after a given dose. So HCG was chosen just as one that there hasn't been a lot of research on in terms of urinary excretion of it. And there's several other drugs that are like it as well that we hope to study in the near future.
Interesting. Well, listeners will have to stay tuned to hear the results of this exciting project. And I know working on research that you're enthusiastic about and passionate about can be one of the big motivators in science. But I guess for you, Geoff, what do you like most about working in the field of anti-doping and maybe specifically working at SMRTL?
So what I enjoy most about working in anti-doping is ultimately how translational all of the research is. A lot of the work we did in graduate school was focused on cell-based assays. We used some animal models as well, and a lot of basic research. And that stuff is without a doubt extremely important. But just as a personal preference, I wasn't as passionate about the basic research aspect as I am about what we do now. So when I look back at the work we were doing in graduate school, the thought of ever doing a study where we could give a drug to a person was completely unrealistic. It was just a dream at the time. And what's great about the research work that we do at SMRTL is that's exactly what we're doing, and how we can ultimately apply the results to testing strategies as soon as the studies are finished. You give X amount of volunteers this drug, and here's what the metabolic pattern looks like. Immediately, we can put those results into a general testing strategy, or we can learn about how to interpret data that we have in the past or positive findings that we may see in the future. So really, it comes down to how translational all of the work is.
Absolutely. And I think you brought up a really good point there that the data that you're generating, the research that you're doing is not just kind of informing what's happening now and in the future for anti-doping, but also looking backwards into the past. I know oftentimes samples are stored for long periods of time and you can go back and reanalyze and kind of make new decisions with the better, more sensitive tools or more information that you have, which I think is really cool.
Absolutely. And sometimes, as we've learned in the past, that new metabolites may be discovered. So what we try to do with a lot of our studies, most of them, is collect as many samples as we can and store them. There's studies that we've done 7 or 8 years ago that we still have the samples from, just in case there comes a day where somebody discovers a metabolite for one of the drugs that we've tested. Now we have a lot of urine samples that have been banked. We can look back into those urine samples to confirm metabolic patterns or whatever is needed at the time.
Very cool. And I know we touched earlier on some of the recruitment challenges that all clinical researchers face, but I guess for you, what are some of the biggest challenges, Geoff, that you're facing right now in your research?
You absolutely hit it with recruitment. That can be very difficult. But right now, as we try to do more and more drug studies with different drugs, one of the biggest challenges we've faced is getting approval for these studies. But this is rightfully so. Whether it's the IRBs, the medical boards, the FDA, getting approval is a big challenge. But the most important aspect that we have to consider with every study that we're doing is the safety of the volunteers. So it's not necessarily a bad thing that this has become a challenge, because this is just an extra level of safety that we know our volunteers will have when they enroll in these studies. A couple studies recently, we've had trouble acquiring drugs as well, whether it be because of where it sits on as a controlled substance or whether it's just not commonly available or specific formulations that we might be looking for. But in the end, we've worked a lot with the IRBs and with the FDA, and they've been quite helpful with these studies as we've had a lot of back and forth with them, taking their advice on how to ensure safety, maybe altered dosing patterns based on what we had initially proposed, or for them advising us to get additional collections to ensure safety of volunteers. So one of the biggest challenges that we currently face is getting approval for some of these studies, but we are getting approval for them. And I think it's a good thing that this is a challenge.
Definitely. I think having that process in place is very important, like you said, for safety. But I guess to give our listeners a sense of the process, what are the steps and maybe what is the timeline that you have to go through to get these studies approved?
So it really depends on the drug that we're trying to study. The steps that we'll do first is design the dosing protocol and how often we want to collect samples and ultimately what we want to look for. We're not just giving drugs to give them. Obviously, there's a goal with every research project. So we need to tailor the dosing pattern or tailor the collections to give us the best chance to reach this goal, to unequivocally answer the question that we're looking for. So we design these protocols and draw up informed consent. And what we do is we submit these to an institutional review board who will review and any additional documentation that they may need, be it licensure, be it safety monitoring plans. They'll review all of this, and they typically will get back to us in about 2 to 3 weeks, which is a really nice turnaround. When we work with the FDA, that's typically about a 30-day turnaround. So ideally, if there were no hiccups and we had everything that we needed to right away, we like to get approval for a project from a regulatory board within 2 to 3 months of the idea conception.
Definitely. You know, science can be a slow process sometimes. So it's good to hear that at least these timelines are more on the order of weeks than kind of months, years, et cetera. So
that's the goal. It doesn't always end up that way. There's one I'm working on now that's probably been 6 to 9 months, but that's just the way it goes.
Absolutely. So we touched on the challenges, but we don't just want to talk about what's difficult about life in your laboratory there. So Geoff, do you have a favorite success story from your career or your research that you'd like to share with me and our listeners?
Sure. A little bit outside of anti-doping, but back in 2020 here at SMRTL, we set up a COVID testing facility, basically from the ground up with a lot of help from our partners. And that turned into a great resource for the community for COVID testing around here to the point where we had analyzed over 1 million COVID tests.
Wow.
But when it comes to anti-doping, if you look at my, albeit short career as a whole, my favorite success story, I think, is how far we've advanced our research program and our research potential since I've been here. So before starting full-time in 2015, we had never done any human drug administration studies here at SMRTL. And in the last 7+ years, we've conducted 16 studies that have involved human volunteers, and 12 of these have involved some kind of prohibited drug or prohibited method administration. That, in my opinion, is a pretty huge success and shows how great of a support staff that we have here at SMRTL and how well everybody works together here, that the teammates we have here are buying into the research and are willing to help out with the projects in any way possible. So that's been, I think, a huge success story. And then if you look at maybe a specific study or a specific example, I think my favorite study that we've done in terms of result output was a clomiphene administration that we did back in 2017 or 2018. We went in with one idea in mind. We wanted to look at the metabolism patterns, and we wanted to look at ultimately how long this drug was detectable after being used, how long it was detectable in urine. And we were able to track patients for almost a year and study their urine. And what we eventually found in the end was these clomiphene and its metabolites were detectable in urine for up to and likely longer than a year. And this finding was unique because there's not a lot of clinical studies that will, first of all, as we mentioned earlier, look at urine, but second of all, that will follow up patients for that long to see whether or not a drug is still present. So that may have been my favorite study that we've done since I've been here.
Oh, that's really interesting. So what is clomiphene? What class of drug are we talking about?
So clomiphene is what's called a SERM. It's a selective estrogen receptor modulator. And typically it's used for females who are looking to induce ovulation. And there is off-label use, and perhaps it's approved. I have to double check on that, but I know use in men for increasing testosterone levels. So what we did in this type of study was we had a goal of looking at how long it was detectable in urine. But as I said, we try to collect as much information as possible from every study that we do. So we also looked at testosterone and gonadotropin levels in blood from that, and saw in a lot of individuals that their testosterone levels were increasing 200 to 300%. And these are healthy men that were doing the study. So you could see why it would be a very good performance enhancing drug if you were looking to increase testosterone. But at the same time, we learned that the drug is going to be detectable for an extremely long time. So assuming you're using it for pharmaceutical benefit, we're probably going to detect it in urine.
Absolutely. That's fascinating. And I know earlier this year, you and your colleagues published a study examining the detectability of growth hormone use when testing using capillary dried blood spots. So can you tell us more about the goals of this study and what you found in that paper?
Right. The goals of this study were to transition standard growth hormone testing from serum to dried blood. Serum that has to be collected from a phlebotomist in the field with a needle in the arm into a serum collection tube. What we wanted to do was try to eliminate that and make it easier for the athlete, easier for the collecting bodies. So what we did in this case, we used a shoulder collection device that collects a small amount of blood that will dry and then can be shipped to the laboratory. Again, the goal of this study was to try to replace conventional GH testing in serum and transition that to dried blood. So what we did with our partners at the PCC and with Major League Baseball, the first step of which was to get approval from an IRB to give a single dose of HGH to individuals and then test essentially their dried blood after the dose. After we did that study, we felt it would be beneficial to give more than one dose. So I can't recall how many we did, whether it was somewhere between 6 and 8 doses to individuals to look at if there was a cumulative effect of GH using it more than once. So those were our positive, I don't want to say controls, but in this overall study, that was our positive control to say, all right, if we know that you give these individuals growth hormone in a controlled environment, what does the dried blood look like after a certain number of hours after dose? And the final point of that was, again, with our partners at the PCC and Major League Baseball, was over the course of a few years, a collection of over 1,200 samples in the field using these shoulder collection devices, shipping them to our laboratory and having us analyze them for growth hormone. And what we found was very simple. Just putting it into words of growth hormone abuse can be detected in dried blood. Whether it can completely replace serum and be as sensitive, that may depend on who you're talking to or what your goal is in your drug testing program. But we answered the question that we wanted to, which was, can you detect growth hormone use in dried blood spots? And we were able to see that.
Very interesting. And as you mentioned, the study was done in partnership with the PCC, but also with Major League Baseball. So what was it like working with a major sporting organization like Major League Baseball for this study?
It was great. So they certainly have the resources to help us out with this project. I believe it was maybe at the end of 2019, we were able to go down to the Arizona Fall League, which is a minor league baseball league, and work with the athletes down there, telling them about the test. And eventually several of them volunteered to have their blood drawn as part of this field study. So it was a cool experience as a laboratory analyst or a lab manager. We don't get to go out into the field very often and interact with the athletes or collect samples in the field. Mm-hmm. That was fun. It was unique. It was a cool experience.
That is awesome. And I know you also recently published work investigating the feasibility of using capillary whole blood collection to analyze athlete biological passports. So, Geoff, can you talk about maybe the potential impact of this line of work on anti-doping testing and some of the longitudinal monitoring that you do?
One of the big hurdles to anti-doping testing for laboratories, for testing authorities, is the cost. There's a lot of labor that is involved with it, and doing science is expensive. So one thing we hope that this capillary work, what we showed was we had several volunteers who had whole blood, so liquid blood collected from capillaries in their shoulder. And we also at the same time collected venous blood according to standard methods as you would for the ABP, for the athlete biological passport. And we compared these two different types of blood, the capillary blood and the venous blood, and found out that they were similar enough that we should be able to implement this into the athlete biological passport. This is kind of an example of a quick and dirty study that I was talking about earlier, where we knew that we didn't need 9 months to a year to do this. We knew we didn't need a whole bunch of money to do this. So this was an optimal opportunity for a microgrant for this project specifically. So we were able to just do this work really quickly, show the similarity between the two types of blood, and publish that. And the potential impact that this may have is ideally we can replace, again, the venous draws in the field, the venous blood draws in the field with this capillary collection method. Now, while you still may have to ship this whole blood cold as you would venous blood, the tube sizes are about 1/10 of the size of the venous blood tubes. So ideally, you'd be able to use smaller shipping vessels and save some money there, save some money on collection end as well. and what that money can be used for, potentially more frequent testing of athletes and more frequent longitudinal monitoring of these athletes.
Definitely. So we've mentioned a couple different types of collection methods. And just to make sure our listeners have everything square in their minds, we mentioned venous blood collection, which is kind of the standard method where you need the phlebotomist, you get a whole tube of blood, this capillary collection method, and the dried blood spots. Can you quickly summarize the pros and cons of each?
Not much is going to beat when it comes analytically to the whole venous blood that you're going to get, whether you go to the doctor's office to have blood drawn or wherever. You get a lot of it, and that's been very well studied over the course of decades. What certain drugs look like. You get a metabolic panel at the doctor's office and you know what your values are supposed to look like in serum. So there's no substitute for that when it comes to how easy it is for the laboratory to work with. But what can be difficult about those venous draws, so I guess a con for this, is that you do have to have a trained or certified phlebotomist that goes out into the field with their venous blood collection equipment, which includes needles, and collects these blood tubes out of your vein and ships them cold to the laboratory. So that can be cost prohibitive for several reasons, being labor for the collections or shipping. These dried blood spots ideally can be shipped at room temperature, so they don't involve the expensive cold chain shipping that's required out of whole blood. A con for the laboratory is that we don't get as much blood. You may, in a whole blood tube, get somewhere between 2 to 3 mils of blood. A lot of these dried blood spots will come in at 20 microliters. So you're looking at a significantly lower volume of blood to be able to test and to be able to work with. So that's certainly a challenge that we're facing right now and trying to overcome.
Well, thank you for giving us the details. And you talked about some of these recent research projects, but I guess what are the future directions that you're looking forward to pursuing with this research?
Especially the near future, we're sticking to a similar theme that we have here with the human administration studies. There's a number of studies in the pipeline right now. And as I mentioned, there's a lot of drugs that just haven't been well studied as to what the urinary output looks like after dosing, what the metabolic pattern looks like, how long these things may be detectable in urine, what the concentrations of analytes may be timing after doses. And all of these things can really, really benefit the anti-doping field as a whole. So right now, we have a number of studies in the pipeline that are specifically focusing on drug administrations and better understanding what the output looks like.
Interesting. And I think this area of research is rife with questions just because new drugs keep coming on the market.
It is. Those drugs are very difficult to get approval for. So we can only do human administration studies for drugs that have FDA approval. There's certainly word that there are athletes using drugs that are not FDA approved, whether they're purchasing them as research chemicals, whether they're appearing in supplements. So unfortunately, there's a lot of those compounds that we want to study, but trying to get IRB or FDA approval for an unapproved drug is extremely difficult. But you're absolutely right that there's so many of these with new drugs that are coming out that we're looking to tackle as quickly as we can.
Absolutely. And we sort of hinted at this in your response here, but oftentimes in research, you're faced with limitations, whether it's concerns about safety, whether it's time, whether it's funding. If we took away the barriers that normally hold you back, Geoff, is there one particular problem or question in anti-doping science that you would want to solve most?
I would answer this in two ways because, as I mentioned, my role in the laboratory is twofold. From a research perspective, the biggest thing that we would like here is we just need to do more studies. We need a bigger team of people. We need more people that are involved in these research studies. We need more studies that focus on controlled administrations, better understanding of, as I mentioned earlier, urinary concentrations of analytes, metabolic patterns. Even more so, it would be great to have complementary blood and urine data to help better understand when drugs are administered. We have ideas for a lot of these. Right now, we just need the personnel that we can run all of these studies as quickly as we can. From the testing end for anti-doping science, I think in general, the most impactful thing, I think especially in the United States, would be to find an efficient and cost-effective way to analyze every sample for what we currently use as a testosterone confirmation method using isotope ratio mass spectrometry. So this is a very labor-intensive test. It's a very expensive test, but the data that you get out of it tells you a lot. If there were an efficient or a cost-effective way to run every single one of the samples that we analyze through this method, I think that would be extremely beneficial. I'm no expert in that field. I just know that that is one area that anti-doping certainly would benefit from greatly.
Definitely. And it sounds like it's this sort of constant battle between cost and time and the amount of information that you need or would like to have, which I think is something that you're always going to face in anti-doping, unfortunately. But I know you're making great strides in advancing anti-doping science there at SMRTL. So if our listeners want to learn more about you, the research and the work there at SMRTL, what's the best way for them to do so?
I think the best way would just be to look up Sports Medicine Research and Testing Lab or any one of our authors, whether it be Dr. Eichner, Dr. Holly Cox, or myself on PubMed. If you search one of our names, you search the company name, that will be the best place to go to learn more about the type of work that we do here, the type of work that we've done in the past specifically, and what we've published for readers to see.
Fantastic. Well, listeners, definitely check out some of the fantastic publications that are coming out of SMRTL. And Geoff, it's been so great to have you with us on the show today.
Thanks for having me on. That was fun.
Well, it's been great to chat. And listeners, wonderful to have you here as well. And listeners, if you want to learn more about SMRTL, you should also check out our interview in episode 16 with SMRTL's president, Dr. Daniel Eichner. And if you haven't had a chance yet, it would be amazing if you could subscribe, rate, and review the show on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.